Hello guys, this is Dr. Daniel Qasim and welcome to sqadia.com. For more details of my profile, kindly visit sqadia.com. So in today's lecture, we're going to discuss the contraction and relaxation of smooth muscle. So before starting or right away jumping to the contraction of smooth muscle, we should know a little bit about the smooth muscle cell. We're going to be discussing the contraction and relaxation of the smooth muscle, but before that we're going to discuss the classification of smooth muscles, the difference between the smooth and the skeletal muscle, the neuronal and hormonal control, and the membrane and action potential of smooth muscle, and in the end we're going to summarize our whole lecture. So smooth muscle cell is an involuntary non-striated muscle. Unlike the skeletal muscle, which is the striated muscle, the cells of smooth muscle are fusiform in shape or spindle shaped with the centrally placed nuclei or centrally placed nucleus. Then there are axons having swellings at intervals which containing the neurotransmitters, and these swellings are called as the varicosities. They are present along the muscle fiber. Unlike the skeletal muscle, in which it has the axon terminal ends which contains the neurotransmitters which are released in the synaptic cleft and they bind on the receptors of the sarcolemma. So that's how the action potential is generated in skeletal muscle, while here in smooth muscle cell there are varicosities containing the neurotransmitters which are usually acetylcholine, which is same as that of skeletal muscle, and epinephrine or hormone which is released from the adrenal medulla, and the signals are passed to perform the action potential or inhibition of or relaxation of smooth muscle cell. So why this spindle shape? What's the function of this spindly shaped smooth muscle cell? It provides the elasticity and it provides a tension for a longer period. So the contraction in the smooth muscle cells, like in the blood vessels, in the bile duct, or maybe in GIT, the elasticity is there because of the shape of the smooth muscle, plus the contraction ability, the capability to perform to last the contraction for the longer period of time. So we're going to classify smooth muscle cell into two types. Number one is multi-unit smooth muscle. So in this type of smooth muscle, each muscle fiber, it contracts independently. It is controlled by the nerve signals. It is mainly present in the ciliary muscle of eye and in pilorectal muscles, causing the erection of hair by the sympathetic nervous system. Then we have single unit smooth muscle, which is unitary, syncytial and visceral smooth muscle. There are millions of muscle fibers. They are contracting as a single unit. There are gap junctions that are present between the cells so that the action potential can be transmitted from one cell to the another. While if this point is taken into consideration, and if you want to compare it with the skeletal muscle, so that we have myelin sheaths, and there is a jumping of action potential along the interval, and the nerve impulse is transmitted in the nerve endings, while here they have the gap junctions between the cells, so the action potential can travel from one cell to the another. The main example of single unit smooth muscle cell is bile duct, uterus, and the blood vessels. So this is a diagram of a single unit smooth muscle. You can see the axon having the autonomic neurons containing varicosities, the swellings, which are also called as boutons. They contain the neurotransmitters. and you can see the action potential. You can see the gap junctions from where the action potential is transmitted from one cell to the other cell, and these gap junctions help in transmitting the action potential throughout the muscle fiber. And this whole muscle fiber cells, they contract at the same time. They contract in a single unit. Like for example, they are present in the bile duct, they are present in the uterus and in the blood vessels, so they all contract or relax at the same time, so they function as a single unit. That is why it is called as single unit smooth muscle. Single unit smooth muscle, which is also called as visceral smooth muscle or unitary or syncytial smooth muscle. Then we have a diagram of multi-unit smooth muscle. In multi-unit smooth muscle, they are the axons or the autonomic neurons. They are forming a syncytium or synapses with the muscle fiber cells. You can see they all contract individually. So if this impulse coming down or the neurotransmitters are transmitted from these nerve endings or these nerve terminals, so only this muscle fiber is going to contract. So like the ciliary muscle of eye and piloerector muscles, and causing the erection of hair. So what are the presence? There are few intermediate filaments and few the cytoskeleton of the smooth muscle cell containing few actin filaments, and there are few myosin filaments with few contractile units, and the intermediate filaments that are present along with the microfilaments, they are present in the smooth muscle cell. So each will be discussed separately. The actin filaments, they attach to the dense bodies and are dispersed in the cell. They are connected with structural proteins and held in place and sometimes linked with the other dense bodies. Few are attached with the cell membrane, forming bonds with the dense bodies of the other cell, and that is how the force is transmitted. As compared to myosin in filaments, these are dispersed along in between the actin filaments, and they are twice the size of the actin filaments. So what are dense bodies? These are the bodies that are present throughout the muscle fiber cell, and they are connected with the intermediate filaments, and these intermediate filaments link one dense body to the another dense body, and they are connected with the sarcolemma, with the membrane of the smooth muscle. The contractile units, they compose of actin filaments which are radiating from the dense bodies overlapping a single myosin filament. So whenever the action potential is generated, it is generated through the gap junctions in the smooth muscle, and once they enter into smooth muscle fiber, the action potential is transmitted, and hence causing the cross bridging or the overlapping of the actin and the myosin filaments, and hence the contraction and the relaxation occurs, and these elements help in transmitting the force. So this is a smooth muscle cell in a relaxed state form. In the relaxed state form, you can see these are the dense bodies and the intermediate filaments. They are connected with the dense bodies of this one dense body is connected with the another dense body with the help of the intermediate filaments, and these intermediate filaments are attached to the sarcolemma or the plasma membrane of the smooth muscle cell, and you can see these dense bodies are dispersed throughout the smooth muscle. While in the contracted form, you can see the length is not changed. It is as it is in the relaxed state, so you can appreciate the length of the smooth muscle. While in the contracted form, you can see the length has been shortened because there is tension in the smooth muscle, and the contracted form, you can see that all cross bridges, they are cross linking with each other, reducing the size or reducing the length of a smooth muscle, and while decreasing the diameter of the smooth muscle, and the function of contraction occurs. So the difference between smooth muscle and skeletal muscle, number one, we have just discussed that smooth muscle is not a striated muscle, and it is an involuntary smooth non-striated muscle, while skeletal muscle, it is voluntary, number one, and number two is it is a striated muscle. The prolonged tone of contractions we can witness in smooth muscle, while in skeletal muscle there is no prolonged tones. There is low energy requirement. ATP is not consumed in higher forms like in the skeletal muscle, because ATP helps in generating energy for the contraction, for the calcium binding in the skeletal muscle, while in smooth muscle ATP has a very little function in generating energy. So cross-bridging cycling between the ATP, between the actin and the myosin filaments, it is slower than the skeletal muscle. There is slow onset of contraction and relaxation after excitation, while in the skeletal muscle, since it is voluntary, so there is faster onset of contraction and faster onset of relaxation. There is an increased force of contraction in smooth muscle, while in skeletal muscle there is minimal force of contraction. So after contraction, once the smooth muscle is contracted, its length decreases for the greater percentage, almost size to zero, because it's all crumbled up. The smooth muscle all crumbles up, so it decreases the diameter of the whole smooth muscle, while 1/4 to 1/3 of the stretch of the length is reduced or stretched in the skeletal muscle. So there is so there are no prolonged tones. That is why there are no prolonged tones or of contractions in the skeletal muscle, while in the smooth muscle, because of decreasing the size to almost zero, there are prolonged tones of contractions. Now, contraction of smooth muscle, latch mechanism. So latch mechanism, what is latch mechanism? Latch mechanism is prolonged holding contractions with minimal utilization of ATP, and the full force of contraction is does occur as a result of this prolonged holding contractions. So as we can see, the latch mechanism is while in skeletal muscle there is no latch mechanism. We go through with the excitation contraction coupling between the actin and the myosin filaments. While by keeping this scenario of skeletal muscle in mind, so it will be easy for us to compare the contraction of smooth muscle with the skeletal muscle. So this is a myosin filament, and it's you can see the head, and you can see the ADP here instead of ATP, means the actin filaments bind with it. So there are no troponin proteins present, which are present in the skeletal muscle for the binding of the myosin head, and contraction does occur after that. But here there are no troponin proteins, and ADP is basically the phosphorylation of the myosin head with the help of myosin kinase light chains, which is the regulatory chain. It converts the ATP, because the initial state is the formation of ATP, and that debinds the myosin head from the actin filament. So once the phosphorylation occurs on this area, there is a conversion of ATP to ADP, and this myosin head then binds with the actin filaments, and there is myosin head rotates towards the center of the sarcomere and actin, and hence the cross bridging of actin and myosin filaments occur, and contraction does occur, and then as a resultant, once the muscle is in relaxed state, so what happens that ADP is then converted to ATP, and the cross bridging is between the actin filaments and myosin filament. They detach from each other. So calcium is an important ion which is present in high percentage in the extracellular fluid, and is also present in the sarcoplasmic reticulum, which is the endoplasmic reticulum of the smooth muscle. So calcium binds with the calmodulin and activates the myosin kinase and phosphorylation of the myosin head. It doesn't, smooth muscle actin filament doesn't contain troponin. There are light chains of each myosin head become phosphorylated, which we have just discussed. There's a binding of the actin filaments, and hence contraction occurs. So when the calcium concentration falls below the normal level, there is a cessation of contraction. There are no calcium in the extracellular fluid and no calcium in the intercellular fluid to be transported from the receptors at the sarcolemma into the cell, or maybe from the sarcoplasmic reticulum. There will be no phosphorylation, and there is no conversion of ATP to ADP. There will be no formation of myosin light chain kinase, and hence the contraction doesn't occur, and phosphorylation of myosin head is usually prevented by the myosin phosphatase, which are present, which splits the phosphate bonds and converts the ADP to ATP. Hence the inhibition or the relaxation after the excitation occurs. So here's a diagram showing the entry of calcium and the phosphorylation of the myosin head and the contraction of the smooth muscle. So calcium ions, with the help of the receptors, with the calcium ions once extracellular, because the calcium ions are in higher percentage in the extracellular fluid, they enter through the receptors at the sarcolemma, and they got released from the sarcoplasmic reticulum. Once the calcium enters intracellularly, it attaches with the calmodulin protein, and once it does, it causes the phosphorylation of the myosin head, myosin light chain kinase. With the help of myosin light chain kinase, the ATP is converted to ADP and phosphate bonds are released, and then it causes the actin and the myosin filaments, which forming cross bridges, and there is an increased muscle tension, and there is a contraction in the smooth muscle is witnessed. So up till now we have discussed what is smooth muscle. Smooth muscle is an involuntary non-striated muscle. It is classified into two types. One is multi-unit smooth muscle, and the other one is solitary or the unitary smooth muscle. Which is unitary smooth muscle is present in the blood vessels, in the uterus, in the bile duct, while the multi-unit, they are present in the ciliary muscle of eye and pilorectal muscle of hair. The main difference between the two is that single unit smooth muscles, they contract as a single unit, while the muscle fibers that are present in the multi-unit smooth muscle, they contract individually. The impulses coming from the axon into the muscle fiber or the neurotransmitter, they individually target the muscle fiber, and hence the response of contraction takes place individually, not as a unit. Then we have discussed the difference between the smooth muscle and the skeletal muscle. So smooth muscle is all involuntary, while the skeletal muscle is voluntary. Plus skeletal muscle is a striated muscle, while smooth muscle is a non-striated muscle having a fusiform shape or spindly shaped, which increases the elasticity of the smooth muscle, while skeletal muscle is not an elastic muscle fiber. Then we compared the prolonged tonic contractions in the smooth muscle, while in the skeletal muscle there aren't any prolonged muscle contractions. there is less energy is consumed in the smooth muscle contraction, while a high amount of energy is consumed in the skeletal muscle contractions. There is a phosphorylation of the myosin head with the help of the latch mechanism, causing the prolonged contractions, while converting the ATP to ADP, and hence the actin myosin cross bridging occurs. While in the skeletal muscle there is a troponin protein, and there is a cross bridging between the myosin and the actin filaments with the help of the ATP generation. So ATP is generated. So these are the main differences. However, the smooth muscle after the contraction is reduced in diameter, almost size to 0, while 1/4 to 1/3 of the entire tension length is stretched in the skeletal muscle, and the membrane and the action potential which we have just discussed is once the calcium receptors present at the sarcolemma and in the sarcoplasmic reticulum, once they get inside of the cell in the intercellular area, they bind with the calmodulin protein, which causing the phosphorylation of the myosin and light chain, which is a regulatory chain. And once it is activated, the myosin head binds with the actin filaments, and it is pulled towards the center of the sarcomere, and hence the cross bridging of the actin and myosin filaments occur. While in the relaxation or form, or in the inhibitory form, the presence of the phosphatase, myosin phosphatase, an enzyme which converts the dephosphorylate the myosin head, and hence the detachment of the actin and the myosin filaments occur. So in the neuronal and the hormonal control of the smooth muscle contractions, there are diffuse junctions, the sites of the neurotransmitter release, that are acetylcholine and norepinephrine or epinephrine. There are the autonomic fibers from the diffuse junctions secreting matrix on the coating of the smooth muscles and diffusing the cells. So there are varicosities that are present in the axons, which we have discussed earlier, and they contain the neurotransmitters. So once the axon innervate a muscle fiber, like we have discussed in skeletal muscle, there are multiple branches that are innervating the skeletal muscle and at the motor end plate, while in there are varicosities that are present in the axons. They contain the neurotransmitters, and they are localized, and they release to the specific part of the smooth muscle. So vesicles containing neurotransmitters, which are acetylcholine and norepinephrine. What acetylcholine is acetylcholine released from the smooth muscle cell with the help of parasympathetic stimulation. They bind on the muscarinic receptors and causing the contraction in the smooth muscle, while the epinephrine, they target the sympathetic nervous system, and they relaxes the smooth muscle cell. So contact junctions which are present in the multi-type. In the multi-type, there are the contact junctions, while in the single unit we had the gap junctions. So that is why they are they contract as in a single unit, while the varicosities that are present, that are lying on the muscle fiber, they release the neurotransmitter, and they causing the release of the neurotransmitter, that is the acetylcholine and the epinephrine. So this is a diagram of a of the autonomic neuron possessing the varicosities and containing the neurotransmitters, which are green in color, and once they are released and they bind on the receptors of the sarcolemma, the action potential is generated from one cell to the another with the help of the gap junctions, and they perform their function of contraction and relaxation accordingly. So the action potential we come across in the resting membrane potential of a smooth muscle is usually minus 50 to minus 60 millivolts, which is 30 millivolts less negative in the skeletal muscle, and that is why since it is less negative, so more threshold is required, and so there is prolonged tonic contractions as compared with the skeletal muscles. We have spike potential, which is common in the single unit type, in which there is electrical stimulation or transmitter release by hormones spontaneous generation. So action potential with plateaus are like typical spike potentials. Having a plateau, having a repolarization is delayed for 100 millions of seconds. So that is why there are prolonged contractions which are seen in the uterus, in the vascular smooth muscle, and it is quite obvious when the fetus reaches the uterine walls, creating a tensions. So that whole contraction by the release of the oxygen, the whole contraction intensifies at the uterine walls, and these prolonged contractions are witnessed in the smooth muscle with the help of the formation of plateau in the action potential. Then we have the slow waves potential, which are common in the single unit type. They are spontaneous generation of action potentials because of the presence of the gap junctions. There are slow oscillations in the membrane potential and the oscillations in the sodium pump activity. So what it does is it creates more negative when pumped rapidly and less negative when pumped slowly. There are conduction of ion channels, and the action potential is generated when slow wave rises above the threshold minus 35 millivolts, since it is minus 30 lesser than the skeletal muscle, and because of these few features in the potential generation, the slow waves, because of these features, there are prolonged contractions in the smooth muscle cell. And then there is spontaneous action potential when the muscle contraction is stressed, as in the gut wall. So any distension once the food enters into the stomach, or once it enters in the duodenum or in the esophagus, there is a distension in the wall of the gut, and because of this stretching, the receptors are stimulated, and the action potential hence occurs by the in association with the slow waves, and there is a decrease in the negativity of the membrane potential. There is spontaneous action potential we can appreciate in the gut wall. So these are the slow waves. You can see there's an action potential, there's a membrane potential, which is a graph from 0 to 70. There's an action potential between 40 to 50. And then we have the spikes. You can see there is a stimulation by the parasympathetic acetylcholine and the stretch. So this hyperpolarization by the norepinephrine and the sympathetics. So these hormones, they basically relax the smooth muscle, and these factors, they activate the or contract the wall of the smooth muscle. there are few factors which can cause smooth muscle contraction without degeneration of the action potential. These can be by the local tissue factors causing the vasodilation in the blood vessels and they relaxing the smooth muscle. There are hormones with the excitatory and the inhibitory response, and they are the source of the calcium ions from the extracellular fluid and the sarcoplasmic reticulum. So calcium pumps removes the calcium from the intercellular fluid to the extracellular fluid, or sometimes into the sarcoplasmic reticulum. We have discussed about the calcium, the role of the calcium, of its binding with the calmodulin and generating the action potential and helps in contraction. So local tissue factors can causing the vasodilation in the blood pressure and in the blood vessels and relaxing the smooth muscle. For example, if the blood pressure is low, so blood vessels usually with the help of the baroreceptors, they send the signals and the signals are sent back to the blood vessel causing the vasodilation, and so the blood flow is maintained and relaxing the smooth muscle, the walls of the blood vessel, and hence the whole blood vessel, because since it is a single unit smooth muscle, so blood can be so it is dilated or stretched or contracted for a longer period of times, and excitatory and the inhibitory factors, which are the acetylcholine and with the help of the parasympathetic stimulation, they cause contraction and the inhibition and the relaxation by the epinephrine. So we're going to go through the whole lecture of today. So first we discussed about the smooth muscle, which is an involuntary fusiform shaped with the centrally placed nucleus and the axons which are innervating the muscle fiber cells possess the swellings, or which we call as varicosities or boutons. They contain the neurotransmitters, which are the acetylcholine and norepinephrine, and their function is acetylcholine function with the help of parasympathetic stimulation. They causing the contraction in the smooth muscle, while norepinephrine or the epinephrine, they causing the relaxation of hormone which is released from the adrenal medulla, and they causing with the help of sympathetic stimulation, they causing the relaxation in the wall of the smooth muscle. Then we classified the smooth muscle into two types. One was single unit and the other was multi-unit smooth muscle. The multi-unit smooth muscle cell is present in the ciliary muscle of eye. They all contract individually, plus they are present in the pilorectal muscles of hair and causing their action by the sympathetic stimulation. In unitary or in the single unit smooth muscle, so they are present. There is a presence of gap junctions in between the muscle fiber, and so that the force is transmitted equally, so they all contract as a single unit. So ATP is transmitted in from one point, the action potential is transmitted from one muscle fiber to the next muscle fiber, and hence the cross-linking between the actin and the myosin filaments occur. Then we discussed about the comparison between the smooth and skeletal muscle. There are prolonged contractions are present in the smooth muscle, while in skeletal muscle there are no prolonged contractions. There is an increased force in the skeletal muscle of contraction. There is a low amount of force is required in the smooth muscle. Smooth muscle is a non-striated, while the skeletal muscle is a striated muscle with the voluntary action. Plus, smooth muscle is decreases its diameter during the contracted form, while 1/4 to 1/3 of the stretched length of the force is for the skeletal muscle. Then we discussed about the latch mechanism. These are the prolonged prolonged contractions with little energy consumption. It is the mechanism was the phosphorylation of the myosin head and converting and forming the myosin light chain kinase, which converts the ATP to ADP, and these ADP helps in binding the head with the actin filaments, and once the cross bridging occurs, then the ADP is converted back to the ATP by inactivating the myosin light chain kinase and detaching the actin filament from the myosin filament, and hence the relaxation occurs. Then we discussed about the role of the calcium. Once the calcium ions are high in the extracellular fluid, or the action potential needs to be generated, the calcium with the help of the receptors at the present at the sarcolemma enters inside the cell and are released from the sarcoplasmic reticulum, with the receptors that are present at the sarcoplasmic reticulum. They are released and they move inside the cell. They bind with the calmodulin protein, converting or activating or phosphorylating the myosin head by converting the inactive myosin light chain kinase to the active form of myosin light chain kinase, and hence there's a binding of the actin and the myosin filaments, and there is an increase in the muscle contraction, and the contraction does occur. So acetylcholine usually plays a role in the contraction, while they're in the relaxation, epinephrine or the norepinephrine plays a role with the help of this sympathetic stimulation. Then these are the neuronal and the hormonal controls of the contraction and of the excitation and the relaxation in the smooth muscle. Then we discuss about the spike potential. We discuss about the action potential with the plateau. Because of the plateau, since membrane potential is 30 times less negative than the skeletal muscle, so the action potential with the plateau helps in repolarization. There are longer periods of repolarization, are longer. 100 times more milliseconds are required for the repolarization, and hence there are prolonged contractions we can see. So there are slow waves action potential, which we have discussed, and in the single unit smooth muscle cell. Plus we have also discussed about the dense bodies that are present, that are dispersed throughout the smooth muscle cell. These dense bodies are connected with each other with the help of the intermediate filaments, which connect themselves with the sarcolemma and helps in transmitting the force from one muscle fiber cell to the another muscle fiber cell. So the anatomy of the physiology behind the smooth muscle cell is while discussing the contraction of the smooth muscle, it is very important to know that the difference between the skeletal muscle as well. Since skeletal muscle, the role of calcium ions is as important in smooth muscle as it is important in the skeletal muscle. Once the calcium enters inside the cell, it binds with the calmodulin in the smooth muscle cell, while in the skeletal muscle, the troponin proteins that are present, they are covering the head of the actin filaments, so they decover the head and they bind and they form the ATP, and hence the actin and myosin filaments cross bridging occurs. Acetylcholine is a neurotransmitter that is present in both the receptors and both the tissues as a neurotransmitter and providing their role. In skeletal muscle, it is more organized because there are receptors that are present at the sarcolemma. Once the acetylcholine that is released from the end plate, they attach with the receptors, and there is an opening for the calcium channels to enter, and the action potential is generated down the muscle fiber. While here there are in smooth muscle, the latch mechanism came into importance, and you can see the calcium once attached with the calcium module, and there is an inactive form of myosin light chain kinase, and that phosphorylates the myosin head, and hence the binding of the actin and the myosin filament occurs. And once it is in the relaxed state, there is a conversion of dephosphorylation of the myosin head, but with the help of myosin phosphatase it converts the ATP back to ATP, and hence, since this whole mechanism requires less amount of energy, while in the skeletal muscle the huge amount of energy is required to perform the contraction and the relaxation, and hence the excitation contraction coupling occurs in the skeletal muscle, while the latch mechanism is followed in the smooth muscle. So the active form is converted back to the inactive form, and ADP is converted back to ATP, and hence there is the debinding or the deattachment of the actin and myosin filaments occur. Plus we have we know that the single unit smooth muscle, like in the uterus, in the blood vessels, they possess the gap junctions. So action potential which is generated from one muscle fiber is transmitted to the next muscle fiber, and hence the force is transferred or transmitted from one cell to the another cell, and they all contracting equally or at the same time. While in the multi-unit, they all muscle fibers, they are contracting individually on the individual basis. So these phenomena usually on this phenomena, this whole contraction and relaxation occurs, plus the hormones, the sympathetic and the parasympathetic role in the presence of the muscarinic receptors that are present at the muscarinic receptors that are present. They help in initiating the contraction process and the relaxation process. So these two mechanisms, along with the latch mechanism, is very has a synergistic role in it. So the discussion of smooth muscle, once you come to realize about the contraction in the skeletal muscle, and you come to realize the role of the contraction of the smooth muscle, both can be compared quite easily, because few of the points which are very necessary. The number one key point is the role of ATP. Since the role of ATP and the energy consumption is high in the skeletal muscle, which is very important, which is the main differentiating point between the two. So hence that is why there are prolonged tonic contractions in the smooth muscle, while there are minimal time, or the contraction occurs in the skeletal muscle for shorter period of time, and hence the length is shortened to almost to zero in smooth muscle, while in the skeletal muscle only the few of the entire tension length is stretched. So we discussed all these topics of the excitation and the contraction and the relaxation of smooth muscle. I hope you guys understood my today's topic, and thank you for watching and listening to this lecture, and keep on watching sqadia.com.